Welcome to this lecture on the fundamentals of genetics. Today, we're going to explore how scientists map the genome using two-factor and three-factor crosses. Understanding how genes are linked and how they cross over is the foundational step in building genetic roadmaps, which are crucial for everything from breeding better crops to tracking inherited human diseases. Let us dive in and see how this works. To start, imagine you are packing for a trip. You always pack your toothbrush and toothpaste in the same small pouch. Because they are physically close together, they almost always travel together to your destination. In genetics, chromosomes are like those pouches, and genes are the items stored inside them. When two genes are located very close together on the same chromosome, they tend to be inherited together. We call this phenomenon linkage. But how do we measure just how close they are? That is where genetic crosses come in. Now that we understand what linkage is, let us look at how we measure it. A two-factor cross, also known as a two-point test cross, is our way of testing just how close two specific genetic items are to each other. In this experiment, we cross an individual that is heterozygous for two traits with one that is homozygous recessive. If the genes are linked, the original parental combinations will appear much more frequently in the offspring. By looking at the less frequent, new combinations, which we call recombinants, we can measure the physical distance between the two genes. This brings us to the mathematical side of our cross. To find the distance, we calculate the recombination frequency, or RF. Conceptually, this is just the percentage of offspring that experienced a crossover event between the two genes. In this formula, R stands for the total number of recombinant offspring, which are the babies with a different combination of traits than the parents. T represents the total number of offspring produced in the entire cross. We divide R by T and multiply by 100. As the physical distance between the genes increases, the value of R increases, which in turn makes the RF larger, indicating they are further apart. Before we move on, let us see this formula in action with a concrete example. Let us say we are studying fruit flies, looking at two linked genes, one for body color and one for wing shape. We cross a heterozygous fly with a homozygous recessive fly, and we get exactly 1,000 offspring in total. We count them and find 410 wild-type flies and 410 black vestigial flies. These are our most frequent classes, so they are the parental types. The remaining flies, 90 black wild and 90 wild vestigial, are our less frequent recombinant types. We need to use this data to find the genetic distance between the body color gene and the wing shape gene. Now that we have our data, we need to isolate the recombinants to use our formula. We first identify which offspring are the parental types and which are the recombinants. It is crucial to only include the non-parental phenotypes here, because these are the ones that crossed over. We take the 90 black wild flies and the 90 wild vestigial flies and add them together. This gives us an R value of 180. This number represents the total number of offspring that resulted from a crossover event between the two genes during meiosis. By isolating these specific flies, we are directly quantifying the genetic recombination. With our R value calculated, we can now find the final genetic distance. We take our 180 recombinants, divide by the 1,000 total offspring, and multiply by 100. This gives us 18%. In genetics, 1% of recombination is mathematically defined as 1 map unit, or 1 centimorgan. So the physical interpretation here is that the gene for body color and the gene for wing shape are located exactly 18 map units apart on the same chromosome. This distance tells us that these genes are fairly close together, but still have a moderate chance of crossing over during reproduction. So, we have successfully mapped 2 genes. But what if we have more? Imagine you are trying to map out 3 towns along a single highway. If you only measure the distance between A and B, and then B and C separately, you might miss crucial details, like whether B is actually in the middle. A 3-factor cross is like taking a satellite photo of all 3 towns at once. It involves mating an individual heterozygous for 3 linked genes with a homozygous recessive individual. This powerful technique reveals not just the distances, but the exact order the genes appear on the chromosome, which a simple 2-factor cross cannot do reliably. You might wonder why we do not just do multiple 2-factor crosses instead. The reason is accuracy. When genes are far apart, multiple crossovers can occur between them. A double crossover essentially flips the middle gene back to its original association, hiding the recombination event. This makes the outer genes look closer together than they actually are. A 3-factor cross captures these hidden double crossovers, allowing us to correct our distance calculations. Without this technique, our genetic maps would be highly inaccurate, and our understanding of chromosome structure would be fundamentally flawed. To account for these hidden events, we use a slightly modified formula for our 3-factor cross. Here, D refers to the genetic map distance between 2 specific adjacent genes. The variable NSCO stands for the number of single crossovers that occurred specifically between the 2 genes we are measuring. Crucially, we must add NDCO, the number of double crossovers, because a double crossover involves a break between our 2 genes of interest as well. We divide this sum by the total progeny and multiply by 100. If the number of single or double crossovers increases, it means the physical distance between the genes is larger, resulting in a higher value for D. Let us apply this new formula to an example, studying 3 linked genes in corn: virescent, red, and brown. We cross our plants and get 1,000 offspring. We identify our largest numbers, 420 and 420, as the parental types. Our smallest numbers, 5 and 5, are the double crossovers, because 2 simultaneous crossovers are statistically rare. By comparing the alleles of the parental type to the double crossover type, we deduce that the red gene is in the middle. Now we need to determine the map distances between these 3 genes. Now that we know the red gene is in the middle, let us calculate the distance between the first gene and the middle gene. We take the single crossovers that occurred specifically between these 2 loci, which are 60 and 60. We must also add the double crossovers, 5 and 5, because they inherently include a break between the first and second gene. Adding these gives us 130. Dividing by the total, 1,000, and multiplying by 100 gives us exactly 13 map units. This means the physical distance between the first gene and the middle gene is exactly 13 map units. Next, we calculate the distance between the middle gene and the third gene. We use the single crossovers specific to this region, which are 15 and 15. Once again, we must add the double crossovers, 5 and 5, because they also represent a physical break in this specific region of the chromosome. The sum is 40. Dividing 40 by 1,000 and multiplying by 100 yields 4 map units. The total map distance from the first gene to the last gene is simply the sum of these 2 distances,
which is 17 centimorgans, representing the full physical span across this chromosome segment.
Let us visualize what we have just calculated. On screen is the final genetic map for our 3 corn genes. The horizontal line represents the chromosome itself. You can see the 3 genes marked in their exact order: V for virescent, PR for red in the middle, and BM for brown. The distance between the first 2 is clearly labeled as 13 centimorgans, and the distance between the second and third is 4 centimorgans. The overarching bracket shows the total distance of 17 centimorgans. This diagram perfectly captures the physical layout we deduced from our cross. To wrap up our lecture, let us review the most important ideas. We learned that 2-factor crosses are great for finding the basic distance between 2 isolated points on a chromosome. However, 3-factor crosses are vastly superior for building a comprehensive map because they account for hidden double crossovers and reveal the exact order of the genes. Mastering these techniques allows geneticists to visualize the physical layout of chromosomes, which paves the way for everything from advanced genetic engineering to tracking diseases. Great job following along with these complex calculations.